Good evening, both of you. We'll initiate the session in 5 minutes. Okay. Good evening, all of you. So, today we are going to discuss another technique in our genome editing approach. Okay. So, till now we have studied zinc finger nucleases and talon, which are designed to specifically target to a DNA sequence and then modify that and edit that. Okay. So, today we are going to discuss CRISPR, which is a breakthrough technology, which is now all the biotechnologists and biochemists are using in the laboratory. So, can anybody tell what is the full form of CRISPR? Clustered red dye electronic. Yeah. Yes, it is clustered interspersed short palindromic repeats. Okay, so this CRISPR is modified or adapted from bacteria's adaptive immunity against the virus. Okay. So it is a part of bacteria's adaptive immunity system. So where inside a bacterial cell, when a phage, which is a bacteriophage, when it attacks, it releases its genomic DNA to the bacterial cell. Okay. So inside that, the bacteria's genomic DNA, the phage's DNA. So some chunk of this viral DNA gets incorporated in the bacterial DNA. Okay, so the virus releases or incorporates its content of its genomic DNA in the bacterial DNA. Okay, and that chunk of DNA is being used by the bacteria's machinery to prevent the infection which is being caused by the same virus again. Okay, so some other virus introduces some other DNA, another one some other DNA, and all gets some chunk of those gets incorporated in the bacterial genome. Okay, so these are these the chunks of the viral DNA, these are referred as spacers. Okay, because in bacterial genome it is found that these kind of repeats, regular repeats, which are palindromic in nature, have been found. So there are several palindromic repeats are found in the bacterial genome. These are the palindromic repeats, and these particular repeats are separated by a certain lengths um, sequences, which are called or referred as spacers, because these are providing space between the each palindromic sequence. So they are referred as spacers. And And when sequence similarity has been observed, it has been observed these spacers sequences are similar to the viral genomic DNA sequences. So that is how it has been concluded that these spacers, which are of no relation or no sequence similarity to the bacterial genome, because they are actually the viral genome pieces which are incorporated into the bacterial genome. So these spacers and these are the palindromic repeats of the bacterial genomes are present. Okay, so what happens? So when the virus attacks, or then there are two proteins which are synthesized inside the bacteria itself, which are referred as Cas1 and Cas2. Okay, so they excise a piece of the viral DNA which gets integrated into the bacterial genome. Okay, similarly for other viruses as well. So now when the same virus infects the bacteria again, okay, and releases the same piece of DNA, then what happens? So these kind of arrangement, these palindromic repeats separated by the spacer sequences are called as CRISPR array. Okay, clustered re clustered regularly interspersed spacer short palindromic regions. These are the CRISPR array. Okay, so then this particular sequence of the DNA undergo transcription. Okay, that is done by RNA polymerase 3. Okay, let me take a slide. Starch. Okay, so with the action of RNA pol 3, it synthesizes the single stranded RNA. So along with the bacterial DNA, this viral DNA will also get transcribed and produces the RNA. Okay, so this is now referred as pre-CRISPR RNA. Pre-CRISPR RNA, which undergoes further cleavage by another enzyme, which is RNase 3 inside the bacteria. Okay, so then the processing of this sequence happens. These RNAs get separated. So as we have, and then inside the bacterial machinery only, we have another protein which is or another segment of the DNA which is called as tracer RNA. Okay, and then that tracer RNA will produce and bind to that which is complementary to the host RNA. So this is a tracer RNA. Okay. So tracer RNA is the one which is produced by the bacteria itself and also bring the uh, Cas9 protein, which is also synthesized by the bacteria and joins this particular. So this is now referred as CRISPR RNA. Okay. So this brings the Cas9 in the picture. Okay. So this binds with the Cas9 protein as well as this CRISPR RNA as well. Okay. And then what it uh, targets this particular sequence to the new virus that has entered, or the viral DNA that has entered the bacterial system. So like in our previous slide. So here the virus has infected this bacteria again, and now this particular sequence will now be ligated with this particular Cas9, CRISPR Cas9 machinery that has been synthesized here. Okay. This is the Cas9, which is having the so this complex that has been formed here. So this particular RNA is the complementary to this particular sequence, viral sequence. So now this entire complex go and bind to this particular guide sequence, and then this Cas9 which function as an endonuclease. Okay, so this endonuclease activity of the Cas9 guide RNA is being performed to make a cut in both the strands of this viral DNA, and that is how it creates a double strand break. Creates a double strand break in the DNA. So these kind of sequences, wherever it is present in the viral DNA, that it's being cleaved by the action of CRISPR-Cas9 system, and that is how the whole viral DNA gets degraded. Okay, so that is the adaptive immunity adapted by the bacteria to get prevented by the infection of the virus that has already occurred in its system. Okay, so that is how the fragmentation happens. So another thing that comes here, that the same sequence is there in the bacterial genome as well, but the bacterial genome doesn't get fragmented by the action of CRISPR-Cas9 system, because in the bacterial so in the viral DNA, when that complementary sequence bind to this bacterial DNA, it also has one very important sequence, which is a 3 nucleotide PAM sequence. Okay, so this is uh, the PAM sequence, which is generally NGG. So it is referred as protospacer adjacent motif, and this one you can also say protospacer. And this is protospacer adjacent motif, which is not present here in the bacterial genome where it is integrated, but it is present in the viral genome. So that particular PAM sequence is very much important for the endonuclease activity of the Cas9. So if the PAM sequence will be absent, then the Cas9 cannot perform the endonuclease activity, and that is why the bacterial genome remains safe from the action of the CRISPR-Cas9 machinery. Okay, so you can see this is the genomic DNA of the bacteria. This is the whole CRISPR RNA along with the tracer RNA, and this is the Cas9. Okay, you can see. So these are the palindromic sequence, palindromic repeats that were present, and these are the spacers or the protospacers which are found here. And here at this particular location, the PAM sequence is being located, which is being identified, and inside this Cas9 endonuclease, there are two particular subunits of this nuclease, which is HNH, which is histidine and arginine rich domain HNH, and another domain is RUVC, so which makes the cut. One HNH makes where the complementary binding is happening, and the RVUC makes the cut where in the opposite strand. So here you can see this is the organization of this particular protein Cas9, which has these major subunits, which has the REC1, REC2 and REC3, and then it has HNH, RUVC domain, and RUVC and REC1, REC2 is joined by this bridge helix, and also PAM interacting domain. So this is the entire structure of the Cas9 protein. So these REC1, REC3 and REC2 are helping in the sensing and recognition of the DNA to the Cas9 protein, and also the structural modification when the binding takes place. These HNH and RUVC are functioning as an endonuclease, and this PAM interacting motif interacts with the PAM region. Okay, so this is the structure, and this technique is being used for many genome editing mechanisms. Yeah, you can see here this is the foreign DNA, this is the target PAM, NGG here, and the spacer sequence, and this is all the CRISPR RNAs. The same thing, tracer RNA, this is the primary transcript, makes a CRISPR RNA, which is joined by the tracer RNA, and this makes a interface. Okay, so this is called the interface, where the viral RNA gets complementary binding with the tracer RNA and the Cas9 complex and makes the cleavage here that will fragment the viral DNA. So same technology has been used here in the studies. So what is being done here? So suppose if you want to make or you want to edit one particular gene in the genome of any genome, any cell, then so this is your gene of interest. So what can be done here is, if you want to make a cut at any particular specific locus, then what you can do, you can uh, make the binding of that protospacer sequence happen at this particular gene of interest at that particular position where you want to make a cut. So the protospacer sequence is the main sequence that leads the Cas9 machinery towards the genomic DNA or the DNA content where you want to make the cut. So it basically guides the Cas9 protein to the gene of interest or the target. Okay, so that is why that particular RNA is also referred as guide RNA. So this is referred as guide RNA. So if you want to make cut at any particular position, you have to design a complementary guide RNA that will bind to that particular locus. Okay, so the tracer sequence will remain constant. So for your tool designing, you can take a vector or a plasmid where the tracer RNA sequence is already there. Tracer RNA sequence is there, and there you can clone or fuse this particular complementary guide RNA. Okay, and then in the same vector you can put this Cas9 protein. Okay, so when this entire vector construct you can transfer it in your organism, in your cells, and there it will express. So the guide RNA and the tracer RNA will express and make this particular complex, which will lead the Cas9 protein to the target sequence upon the complementary binding of the guide RNA to the targets. And then the guide RNA will make a cut, the PAM locus in the opposite strand. Okay, so this will create a double strand break in the genome. Okay, so this is how the double strand break is being made. So then followed by the break, you can do any modification that you desire. As we have already discussed, the HR machinery can be utilized here in order to insert any particular sequence which is having the homology with the broken ends. Between the broken ends, between these ends, then via the HR machinery, at the genomic locus also it will get integrated. You can stop me if you are having any doubt in understanding. So this is the mutated copy or any mutation or any extra sequence that you want to incorporate at that particular genomic locus, you can add. And similarly, if it is led to non-homologous end joining, it will lead to indel, like insertion of any segment or deletion of certain portion. So that is how also it can be edited. So so till now, is there any confusion? Can you ask any question if you are having, or is it clear to everybody? Okay, if there is no any question. Okay, so these Cas9 or CRISPR-Cas systems are divided into 4 classes. Okay, there are class 1 and class 2, and there are 4 types, type 1, 2, 3, and 4. Okay, so in type 1, so the Cas protein differs in different classes. So in the type 1 you have Cas3, in type 2 you have Cas9, in type 3 you have CSM3A and CMR3B, and in type 5 you have Cas12A. Okay, so both type 2 and type 5 are being extensively used in the genomic study, where you need the effector is the CRISPR RNA and the tracer RNA. And in type 5, you need the effector of CRISPR RNA. Okay. here the PAM sequence in the type 2 Cas9 system is the NGG. So N means any nucleotide and GG are the two guanines. Okay. So as this particular machinery is adapted in such a way that it will cleave the viral DNA. So in the viral DNA, the frequency of getting 2G together is very high. Okay, so this is such a viral type genomic DNA type of uh, analogy. Okay, so this is NGG. So this frequency is very high. However, in bacterial DNA, the frequency of getting GG is very less. So that is why this is a very important specific recognition sequence for the viral DNA and NGG, where N could be any nucleotide followed by the two G's. So it could be G, A, T, C, anything. Okay. So, and here in the type 5, and you can see that there are different type of PAM sequences. For example, Y and T. Y means any pyrimidine, then it is T, thymine, and then N is any nucleotide. Okay, for the recognition. Similarly, another one is also there. Okay, so type 2 produces the double strand break at the exactly same opposite direction, and that is why the break is it is not sticky, it is a flat end. Okay, or it is a blunt end. Okay, it produces ends like this. Okay, however, the DSPs produced here is sticky end with 5 nucleotide protruding. Okay, so suppose if you want to generate the overhangs in case of, so that overhangs can be generated with the help of Cas12 machinery. Okay, so this, like for example, 5' overhang you want to generate so that you can perform certain HR or any type of reactions. So that type of overhangs can be generated when adapted with the Cas12 system. Okay, so is it clear till now? You can ask me because it will get further complicated. Okay, you want to ask me anything, stop me now and ask me, because it is supposed to be an interactive session. So you understood the part of this till now. Is it clear? Okay, so let me ask questions to you then. So, so what is a PAM sequence? What is a PAM sequence? Can anybody tell? It is a protospacer adjacent. So what is the function of PAM sequence? Why it is there and where it is located? At the bacterial genome or at the viral genome? Where? PAM sequence. Yes, and why it is present there? What is the function of PAM sequence? Sequence is the sequence which induces the endonuclease activity of the Cas9. Okay, so endonuclease means no, let's do it. Okay, so the PAM sequence. So PAM sequence. So we know that this particular Okay, so nuclease means to cleave the DNA. Okay, and endonuclease means cleave the DNA internally. Okay, so one thing is DNA is present like this. Another thing is DNA like end of the DNA. Okay, so if the nucleases makes a cut here, like the exposed DNA, then it is called as exonuclease. If the DNA makes a cut in between the nucleus makes a cut in between the DNA, so that is an endonuclease. Okay, so the Cas9 has an endonuclease domain, which needs which will be activated once the Cas9 gets bind to the PAM sequence. Okay, so if the PAM sequence is absent, then that endonuclease activity will not happen. So that is why in the bacterial DNA, the PAM sequence is absent. So even though the protospacer or the guide RNA will go and bind to the bacterial DNA, it will not make a cut because endonuclease activity will not be activated until unless the PAM sequence recognition will happen. Okay, so the PAM sequence recognition will happen only in the viral DNA because there only the PAM sequence exists. Okay, so there only the DNA gets cleaved and fragmented because of the presence of PAM sequence. Okay, so that is why when for our experiment also, I'll give you the example. Okay, so suppose, so if it is clear, the CRISPR system is clear, I'll give you an example. Okay. So suppose you have in your particular experiments, or suppose if you work in a lab and you were told to make a knockout of a particular gene. Okay, so this is your gene of interest in the bacterial machinery. Give me a minute, please. Yeah. Okay, so suppose you have told that in certain organism, for example plasmodium. Okay. Okay, so there is a gene name given to you. For example, you have given that there is a gene named NAD51 and you have to create a double strand break here, or you have to make a knockout of it. So for knockout making, what is the strategy we can do here is you need to have an HR, right? So this is some defective gene. Then this is the homologous to this cassette you provide, this particular template you provide in the cell, and with the homologous recombination event HR. So the replacement of the products will happen. So your gene or the wild type gene will be replaced with the faulty copy. Okay, the faulty copy of the gene. So HR will only initiate once there is a break. So to create a break at one particular in your gene of interest, you have to have this CRISPR mediated break. So suppose this is the gene you have. Okay, so in plasmodium one particular gene you have told, so you go to the database, you find out the gene ID and the sequence of the gene. Okay, so this is the gene sequence which I am showing you. So this gene, so we know the gene starting is always ATG, which is codes for the methionine, and this is the stop codon, which is TAA. Okay, and this is the entire gene sequence. Okay, So now what you can do, you need to design a guide RNA which will be uh, go and bind to this gene of interest. Okay, a guide RNA which will bind to the gene of interest. Then the Cas9 will sit on it and make a cut. Okay, so this is the guide RNA which will guide to the Cas9 to this particular locus, and after the binding, so it will make a cut here and in the opposite strand. So then it will lead to the a double strand break. Okay, a double strand break will occur after the guide RNA mediated Cas9 machinery. Okay, so first step is to design a guide RNA. Okay, so what you do, you paste this particular sequence on a online software which is referred as Chop Chop. Okay, so you have an online software which I can show you. You can just find Chop Chop and write, and on the Google you'll find this Chop Chop. Here you need to add the gene ID of the target that was given, and then you'll give the organism, you'll write the technique and also the for the reason that you want to do it, and then you press find the target site. Okay, so this particular software will give you a list. Okay, it will give you a list which is showing the target sequences of the guide RNA with the efficiency. Okay, so you can see there are several guide RNA sequences are here given, and you can see that there are PAM sequences also given. Okay, NG, GGG, AGG, AGG, TGG, GGG. Okay, so the sequence, as I told you, it's supposed to be NGG, the PAM sequence. Okay, so this is how, and it is also giving you the GC content. Okay, it is showing you that the how much GC content this particular guide RNA sequence holds. So this is 25% GC. Here 30% GC is there. Okay, and again, how much is the efficiency of this guide RNA? Okay, that means like how efficient it will be to use this guide RNA to make a cut at your desired locus. Okay, so this is your gene of interest, and here it is different. So it will make a cut here. Okay, the second one will make a cut here at this particular position in the gene. Okay, uh, and you can also compare the efficiency. Okay, so and then the first one, which is the highly efficient, is the to make a cut here. So that also you can decide where you want to create a cut. Okay, so suppose your particular gene of interest had certain, what to say, um domains which is specific for some certain functions. Okay, so that is how you can decide the locus where you want to create a break, in the N terminal portion, in the C terminal portion, or in the middle portion, and that also you can see the efficiency. Okay, so like here you can say this is the one. Okay, so the sixth one which is having 58% efficiency. Okay, and the third one, okay, which is having 60% efficiency. And here is the, so if suppose this is the domain and you have two options or many options of the guide RNA, you have 27, the third one which is 60%, and you have first one which is 73% efficient. So obviously you will choose the first one if you want to make a more efficient cut there. Okay, so that is how you will get your guide RNA sequence. Similarly, based on the sequence or the target sequences you found there, you can come and compare here that where are those located. So all of those targets, so one of them I have marked here. In all of those targets, I have marked this one, Search. The first one I'm not able to find anywhere. Maybe it is located somewhere, but suppose if this is the guide RNA sequence that you have got, so what you all you can do, you can synthesize an oligo or a DNA or a RNA that will be of this particular sequence. Okay, once you synthesize that, you can clone that particular sequence in a vector. Okay, you can clone this particular sequence here in a vector. So this will be your guide RNA sequence in a vector, and which will be accompanied by tracer RNA sequence. Okay, and the same vector you can clone the Cas9 protein. Okay, and when in the particular organism you'll introduce this particular vector, that time this guide RNA or this particular DNA will produce RNA, and that RNA will go and bind to your gene at this particular locus, and then the Cas9 endonuclease will make a cut there. Okay, so that is how you can use the CRISPR-Cas9 method for the genome editing. Okay, so there are also more application to the CRISPR-Cas9 system. So one is, just a minute. Yeah, so there are several modification also be done for this technique, which is people have made this catalytic inactive Cas9. Okay, that means mutation in certain domains which are like endonuclease domain of the Cas9, which makes the Cas9 catalytically inactive. That means the nuclease activity is lost. Okay? So the nuclease activity is lost because of the several point mutation or certain point mutation that has made in the Cas9 machinery. So that is referred as D-Cas9 or the dead Cas9 because of the absence or the loss of the nuclease activity. Okay. So this catalytically dead nucleus dead Cas9 cannot perform or cannot break the DNA. However, it can still go to the DNA. Okay, so if a guide RNA is provided, with the help of guide RNA, it will be directed to that locus, but it will not break that locus. Okay, so this particular mutant has also been a lot of use to our editing machinery, where so this guide RNA which has guided this catalytic dead. So this is the mutation that has been made, D10A. That means this particular amino acid D has been replaced to A at the 10th position. Okay, that made it catalytically dead or nucleus dead mutant. Okay, along with Cas9, people have fused the cytidine deaminase. Okay, so this is another protein. You can make the fusion protein in frame with each other. So along with the Cas9, if you can make the cytidine deaminase, so this cytidine after the deamination, that means removal of the amino group, it becomes uridine. Okay, so from C to U. Okay, and when it gets converted to the uracil or uridine in the next round of replication, what will happen? So as the Cas9 goes here, it is catalytically dead. It will not make a break, but it will just, with the help of the another enzyme, which is cytidine deaminase, but make a or make a conversion of the from C to U. And when it gets converted from C to U in another round of DNA replication, uh, this particular DNA will now be corrected by the mismatch repair machinery, or so that will be UA pairing, which will be uh, converted to TA. So now the original CG pairing has been converted to TA pairing, okay, which can cause any point mutation. Okay, suppose if your particular code of amino acid which, or the genome or the codon which codes for any particular X amino acid. If this is changed by this particular machinery, it is changed to TAT, then it will code for another amino acid. Why? So then this point mutation can change the structure. It can make it again the loss of mutant, gain of function mutant. It can make or mutate the protein or gene of your interest. Okay, and then you can obviously study its function. If it is not uh, repaired, and then also another path it can also go where this U is being automatically removed by the proofreading machinery of the organism, and then it can also be happen that again get corrected by CG pairing, or the G can also be removed, and that will also be again change in the ORF that will be disrupting the gene of interest. So that is how you can utilize this catalytically dead domain. And also what you can do here is you can also fuse this sgRNA dependent with a GFP protein. So this Cas9 protein, as it was fused here with this deaminase protein, you can also fuse it with the fluorescent protein like GFP. So when this particular protein will be guided by a guide RNA to the chromatin, it can show us, or it will help us in the live imaging of the cell, both in the spatial and the temporal. So the T-Cas9 mediating imaging allows visualization of chromatin in live cell to study the temporal and spatial behavior of the genome in the living cell. Okay, so that is how you can identify. So this was all. Let's go to the question answer. Ask me if you are having any confusion or any doubt. So which of the following? So this particular Cas9 protein, I have told it has been divided into two classes, class 1 and class 2. So the class 1 has three types, type 1, 3, and 4. And the class 2 also has three types, which is type 2, 5, and 6. Okay, so type 2 and type 5, which includes the Cas9 and Cas12, Cas9 which makes the blunt ends, and Cas12 which generates the overhangs. So these are the two types, and these require the CRISPR RNA or a guide RNA mediated recognition or direction. Okay, so these are the types. So there also this 1, 2, and 5. It can recognize and cleave DNA. These three types, type 1, 2, and 5. These Cas9, Cas12, and the type 1, which is also Cas3. These can recognize and cleave the DNA as it is being utilized, as I have shown you till now. Also, other types are there. For example, the fourth one, it can cleave only RNA, not DNA. Okay. So, and also there is type 3, which can edit both RNA and DNA. Okay, so this is the different types of Cas protein. Okay, so if there is option, which of the following is true about the function of Cas protein? So first is type 1, 2, and 5 systems recognize and cleave DNA. Is it correct? Is it correct? Try to answer, please. Unmute yourself and answer. Anybody can answer? Pooja? Yes, what will be the answer? The first option, the type 1, 2, 5, they can recognize and cleave DNA. So these Cas9s are basically a nucleases. That means they will break the nucleic acid. Okay, so in there are categories and there also which will define the specificity. So type 1, 2 and 5 class or type of Cas proteins can recognize and cleave the DNA, which comes under nucleic acid. The type 3 of the Cas protein, it can cleave both RNA and DNA, and the type 4 can only cleave the RNA or can only edit RNA. Okay, so the first option, type 1, 2 and 5, recognize and cleave DNA, is it correct or not? Correct. Yes. So type 6, it recognize and cleave DNA. No, type 6 doesn't. Type 3 can edit RNA. Is it correct? It is correct. Yeah, it is partially correct. That is why it cannot be the correct option. So it can edit RNA as well as DNA. And type 1, 2 and 5 system edit both DNA and RNA, which is incorrect because it cleaves only DNA. Okay, and in this type 2 and type 5 only, our specific Cas9 and Cat12 falls under this category, which is extensively being used in the biotechnology laboratories. Okay, question C. So I have shown you that table. So which of the following is not correct match about the signature protein of the CRISPR-Cas system? Okay, so we have types 1, 2, 3, 5 and 4. We can put it in the same one. This table, it was there. This was the table, right? I don't know. I think there could be a confusion. It is type 5 or 4. I need to cross check that. But till now, you can say that type 1 will be Cas3 or Cas3 prime. Type 2 will be our, which one? Cas? Yes, Cas9. And I have to cross check it. But either it will be type 4 or type 5. It will be Cas12a. Okay. In Cas3, we do not have the Cas12. It is another protein which is listed here. It is CMR3b and CSM3a. CMR3b, CSM3a. Okay. So the which is not a correct match. So which will be the correct option here? So first one, type 1 with Cas3. Correct, right? And the second one, type 2 Cas9. Is it correct? And type 3 Cas12a. Yeah. So the correct option for this question, which is not a correct match, will be this one, C, because these all are correct match. Type 4 will be class 12A. So next, which of the following is not part of 7 domains of uh, Streptococcus pyogenes Cas9? So the Cas9 protein. Okay. So which of these is not a part of this protein? Can you remember? Rec 1, Rec 2, Rec 3 are the part of Sorry, rec? Which one? Ye- Yes, this is not a part of the Cas9 domain. It has rec 1, 2, 3. Okay, it will be correct. It has a bridge helix, which is correct. RUBC, RUBC. Okay. So, the RUBC one is the one which performs the endonuclease activity. Okay. Okay, so there are, I told you like first, so there is an expression of the guide RNA, and then the adaptation. So adaptation process means the insertion of new spacers into the CRISPR locus. So the in the beginning of when the Cas1 and Cas2 creates a cut or degraded, it will go and integrate into the bacterial genome. So that means insertion of a new spacer into the CRISPR locus, and that is called the adaptation step for the CRISPR-Cas9 immunity. Okay, so this will be the correct option. Which of the following is not an example of epigenetic modifying enzyme? Okay, so as we have mentioned here, that cytosine deaminase, right? That the catalytically dead with the dCas9, we can fuse with also with any other epigenetic modifying enzyme. Okay, so these are the epigenetic modifiers which, when directed towards a promoter of any certain gene, which can either methylate the histone or demethylate the histone, and that methylation and non-methylation, which can be directly correlated with the expression or the inactivation of the gene expression. Okay, so that can kind of genetic manipulation can be done when you can fuse the dCas9 with the epigenetic modifiers. Okay, so the first one, so which is not an example of epigenetic modifier. So first one is histone demethylase. So histone demethylase is the one which can demethyl or remove the methyl group from the histone. It comes under one of the epigenetic modifier. Then you have methyl transferase. Okay, H is for histone methyl transferase. Similarly, you have DNA methyl transferase, that is DNMT. So these are also the transcriptional regulators. Then you have histone acetyl transferase that transfer the acetyl group to the histone. That also works as a transcription uh, regulator. These modifications work as a transcription regulatory modification. But the D-acetyl methyl transferase is not one of them. It is not one of the epigenetic modifying enzymes. So this is the wrong option here. Okay. So the TCAS9 mediated imaging allows visualization of in live cells to study the temporal and spatial behavior of, which will be the correct option? Visualization of what? Enzymes or chromatins? No, like you see, genome is the entire DNA of the any cell. Okay, so if this is the cell, the entire DNA content will be the genome. Okay, so this is the genome. But if you are talking about a certain part of that genome, then it is a maybe a chromatin locus or gene or anything. Okay, so those two are two different words. You cannot entirely visualize the entire genome because the CRISPR or the Cas9 will mediate or guide to any particular locus. It will be guided to a particular locus. So if it is guided here at this particular chromatin, so this particular chromatin will be visualized or fluorescent with the help of another fluorescent protein, for example, GFP. So it can allow visualization of chromatin. Yeah, chromatin and the temporal and spatial behavior in the live cell of the genome in the living cells. Okay, so what nucleotide composition of sgRNA is particularly essential to determine its efficiency? Okay, so as we saw that in that list when we in the chop we got pasted the gene ID or anything, then we got a list. So along with the list, we also got a table of the GC content, right? So this GC content is very important. Okay. Okay. You can see the GC content is given here, right? So the GC content is one of the particular or important factor that can tell the efficiency of the this guide RNA, but it is not directly correlated. Okay, it is not directly correlated. Like suppose if you have higher GC content, the binding will be more efficient, the recognition will be more efficient. It is not entirely correlated, but because you can see, look here, the GC content is 40%, still the efficiency is 48%. Here the GC content is only 25%, but still the efficiency is 73%. Okay, so there are certain what you say phenomena and observation that has been made. Okay, like for example, at the 16, like it is a total 20 nucleotides amino acid or nucleotide sequence. So at 16 nucleotide, if you have C, then it will be not that efficient. It's supposed to be G, then that guide RNA will be more efficient. Okay, so these kind of um, uh, observation has been made, and based on that, the guide RNA efficiency has been decided, or it can be decided. But the GC content will play a major role in determining how good the guide RNA is, because so if this is the entire, okay, the guide RNA, right? So here at the last of the uh, towards the PAM sequence, right? Suppose here you have the PAM sequence, here you have the PAM sequence, suppose. So near the PAM sequence, this particular 3' end of this guide RNA, which is called seed, the seed segment. Okay, so this seed segment should be GC rich, but there are certain specific position. For example, before the PAM sequence, the 18th and the 19th nucleotide should be G, then the binding will be more efficient. Okay, so GC content is one of the determining factor, but it is not directly correlated to the efficiency, but it is determined because GC has a triple bond, like the GC bonding is a triple bond hydrogen bonding. So if your guide RNA attached to a triple bond compared to the double bond, it will be more specific and strong bind. So the composition of sgRNA is one of the determining factor is GC content, but not the AT content. That doesn't matter, but the GC content is important. Okay, so what are the effects of U-rich seeds in the context of any guide RNA design? If this particular segment is U-rich, okay, it is U-rich. Suppose here you have the PAM sequence, then you have some sequence like this, U-rich, then there are chances that this particular, so first thing, it is not GC rich, then the binding will be not specific and not efficient, and also if there are more U present at that particular locus, there are chances that it will form a stop codon and terminate the guide RNA expression. Okay, like you are giving this sgRNA in a in a plasmid, right? So when it's supposed to produce the RNA, so but here if you have more U, that could make a stop codon also. Okay, so this seed segment should not be U rich, otherwise if it will terminate the guide RNA or the transcription of the guide RNA. Okay, and also one more thing I would like to point here, so that sequence that you have seen in the list, that is with the PAM sequence. Okay, that is given with the PAM sequence. Okay, that is given with the PAM sequence in the list also, and here also you will see. But when you do this particular cloning, in this vector, you have to remove the PAM sequence. You have to clone it, or you have to put it in the vector without the PAM sequence. Otherwise, when the Cas9 will express and the guide RNA, whatever it will be made, it can target its own thing only. It can target and degrade this guide RNA only if the PAM sequence will be present here. So just like bacterial protospacer sequence, your guide RNA which is present in the plasmid or the vector that you are providing should not contain any PAM sequence. Okay. So the question was, what are the effects of U-rich seeds in the context of gene editing using CRISPR Cas9? So it is the sgRNA transcription. So the transcription will be affected. So the guide RNA, the CRISPR-Cas9 is responsible for. Tell me the answer. Targeting the specific DNA sequence. Yes, that's the correct option. These are the questions we had for today. You are having any doubt, please ask me. Anything you didn't understand, if any part which is confusing, please ask me. One of the previous questions, the RNA can answer the like I think that it should protect the RNA. Why translation? You look, first thing is, look, you are giving it in a vector, right? And this you are giving in a DNA form. Okay, this is because you are giving a, what to say, double helix, okay, and double stranded, okay. Then via the transcription, it will produce the guide RNA, right? Single stranded guide RNA. Okay, single stranded guide RNA, which will be in frame with the tracer RNA also, which is either before it or something. Okay, so this is associated with the tracer RNA after the transcription. Okay, that is how. So from the double stranded DNA, it will go to the via transcription process. It will make the guide RNA, which is an RNA. An RNA is a single stranded, and it, yeah. So it is transcription, and it is now not going to make any protein out of it. It is just an RNA, and it is going to bind to the target site and then cleave the, right, with the Cas9 machinery. It will be cleaved. Okay, so that is why it is sgRNA transcription. Check. Yeah, the RNA transcription, which will be affected, or the uric acid will affect the transcription of this guide RNA. Okay. Anything else? Anyone? No questions. We'll conclude this session. Okay. 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 Good week. Bye bye. I'm closing the session.